Our next speaker is Dr. Daniel Ives. He received his PhD from the University of Cambridge, where he focused on the role of mutations in the mitochondrial genome in rare diseases, embracing computational biology to discover new interventions. In 2017, he founded Shift Bioscience, and since then, he's been on a scientific journey from mitochondria through mouse aging clocks to single-cell aging clocks for CRISPR screens, and finally to now, leading a team that's pioneering machine learning methods to unlock cellular reprogramming for safe reversal of aging and age-driven diseases. So let's welcome Dr. Ives.
Thank you. Thank you. Firstly, thank you, Kristen, for the invitation. I tried to come last year, but there was a last-minute hitch. Fortunately, a year's passed. It's a much more interesting story, so you guys get to have that. I've accidentally forgot to remove my financing seed plus term from this deck. This isn't a financing pitch. So let's just start with what is cellular reprogramming in the title. So this is a paradigm that we can use in the dish to reverse the age of cells back to a biological age of 0. But this is tied up with a lot of dangerous biology that can sort of create cancer in certain circumstances. So we're trying to get rid of the cancer, which seems like the obvious thing to do. So I'm just going to tell you sort of the story of what we've done and how far we've got with that. So just to begin with, rejuvenation, is this really on the table? And just to give you a very powerful example, once upon a time you were a baby, and before you were a baby, you were an individual cell. And that cell started from day 0, even though it came from your parents who would have been in their 20s or 30s. So somehow that cell went back to day 0. And so we don't know if there's a preservation before, like in gamete formation or something in conception or a rejuvenation event. There's certainly evidence. There's all of those things going on. But if we didn't have this mechanism, you'd inherit aging from your parents and have a shorter lifespan, and you'd give your children a shorter lifespan. So it's very promising that nature had to solve this problem very early. So we got a baby and a baby orca. And better than this, we can actually find a set of 4 genes called Yamanaka factors after their sort of discoverer, Shinya Yamanaka, a Japanese scientist. So if you look at the blue line, which is Horvath's clock, Steve Horvath's clock, after just a few weeks, you can take, I think in this case, a 62-year-old female fibroblast and reduce it to a biological age of 0. In the process, you're also increasing something called the pluripotency pathway, which is a sort of cancer-causing pathway. But this is really exciting. It's only 4 genes. We put these 4 genes together, and we can reverse biological age. So we've really got something by the tail here. Obviously, it's not perfect, but the obvious next step is let's make this better. The reason we want to make it better is we don't want this going on. So this is a mouse, obviously not a very happy mouse, but these 2 growths are teratomas or multi-tissue tumors that shouldn't be in that animal. So this is sort of the reality of using these Yamanaka factors without absolute precision. We do have that precision in the dish, but when we start putting these factors into animals, we don't have that type of precision at the moment. So the obvious next step is what is even going on here with these factors? And if we learn the details, can we get rid of this cancer-causing problem? There's some examples. We can separate this cancer-causing problem away from the rejuvenation pathway. So firstly, there's something called transient reprogramming. So we just apply the Yamanaka factors for a couple of days, and we could take an aged fibroblast and sort of move it towards a stem cell state, but not all of the way. So it sort of forgets it's a fibroblast in those 2 days, but when you release the factors, the cell goes back to being a fibroblast. It remembers what it used to be, but in this case, it's actually about 30 years younger. So you can lock in this rejuvenation benefit without transitioning to a stem cell. So that's exciting. You can also take an aged fibroblast or an old fibroblast and directly convert it into an old neuron. So you can change the cell type without affecting sort of the aging component, and this suggests there's a compartmentalization between cell type and aging, which suggests that we could move one without the other, so that's also encouraging. So I just want to highlight the breakthrough that was the epigenetic clock or the DNA methylation clock, or Steve Horvath's clock. Before this clock was discovered, if we were trying to investigate aging biology or investigate things that could reverse aging, we had to do these really long mouse lifespan studies that take 2 to 3 years to complete. And the great irony is, if you try to work in the aging space and ask a reasonable amount of questions, you become very old in the process. But with the aging clock, we can just take the measurement in a relatively short amount of time. We can get a readout like, is this aging biology? Can we rejuvenate? We can do lots of experiments relatively quickly. So I'm not going to go into the details of this clock. That's been relatively well covered. But one room for improvement with this clock, so, I'm the biggest fan of this clock, so I'm definitely not one to criticize. But if we're trying to find safer ways to rejuvenate, we want to do as many experiments as possible, because it's such an exciting outcome if we can do that. So it would be great to have higher throughput. So the epigenetic clocks, we can do a decent amount of experiments. But if we want to discover lots of new targets and lots of drugs that can target those targets, we need a high throughput clock. So this is something we've been working on for quite some time. And this is a breakthrough we had back in July, so not that long ago. To enable this breakthrough, we had to create what we think is the largest single-cell data set for human fibroblasts. So just explain what we did. We took 110 fibroblast samples. So these are fibroblasts from individuals of different ages, 12, 14, 16, all the way up to about 80. We then do single-cell gene expression analysis, so we know what the genes are doing in every single cell. We do methylation analysis, so we can measure the clock in all of those different samples. And then we basically apply machine learning, and the machine learning allows us to find a set of genes that predict the methylation age, but in single cells. So we can take a single cell now and say, what is the methylation age in that individual cell? So this graph just behind me, we are correlating our high throughput clock or single cell clock with what we deem the gold standard, which is the methylation clock, and you can just see there's a high correlation. Yeah, so it means that we can be very confident in our predictions. So what can we do when we have this single cell clock? What's so exciting about that? The first thing is that we can do loads of experiments, right? We can do loads of experiments to find things that are aging the cells and rejuvenating the cells, like try and find something that's better than the Yamanaka factors. We can also take a library of approved molecules, so there's drugs that are out there that are approved for diseases. We can now use this clock to see, do these drugs rejuvenate? We've never been able to, to see whether we could do this or not. And with this clock, we can actually say, look, did this drug actually reverse the age of the cells? And we can also look at these Yamanaka factors, like where exactly is the rejuvenation happening? And just some details on the data set. Not that you were asking, but it took a lot of time and money, so I feel like I need to get something out of it. So we take 61 samples, and then we apply the machine learning to those samples to create these clocks, and then we test those clocks on a separate, sort of independent data set to prove to ourselves that they're as accurate as we want them to be. And when you plot all of the individual cells and you sort of group them together by the similarity of their gene expression, actually create this sort of wonderful picture. I don't know what it is. It sort of looks like a dog to me, but maybe it looks like something else to you. It's like looking at clouds. But every single dot costs about $1.50. So that's about a $1.5 million data set. But this is what we need to do, right? It wasn't just to make a nice picture, but again, I want to get as much value out of that data as possible. So use one, and this is the highest leverage use of this clock. So I'm not going to go into the details of that cycle, but the Yamanaka factors, what are they? They're particular proteins called transcription factors. So transcription factors are very high up in the hierarchy. They're like the management of the cell. They tell other genes and proteins what to do. So Yamanaka factors are they are transcription factors. So if we're looking for a safer way to rejuvenate, we're actually going to look in the universe of transcription factors, which is about 1,300. And there are 365 million combinations of free transcription factors to test, which is just enormous. If we try to test this in the real world, even with our clock, even with our high-throughput clock, it would take about 50 years and $10 billion, and that's just too long. I mean, that's too long for me. Maybe others would take that amount of time. But we can actually use this clock with something called in silico models or cell simulations to actually test up to 500 million interventions. And the idea is, here's all of these combinations, and we're going to try and map the landscape of safe rejuvenation. So, what is the highest peak? So what is the combination of genes that does that the best? Like, what's Mount Everest? What's K2? What's Table Mountain? We want to basically map the whole landscape, and then we can find the best possible starting point for a drug. So, reduce the problem from about 365 million combinations to something between 10 to 1,000. So this is what we want to be able to do. And I sort of talked about these cell simulations, so just briefly touch on that. You almost certainly have heard of ChatGPT. So this is the application of transformer models to language, and you create these amazing chatbots. But you can use the same models to learn the relationships between genes and individual cells, and basically create an abstraction of what's going on in the cell. And you can basically create a simulated version of that, and you can run your experiments in that simulation. And this simulation actually has a meaningful connection with the real world. And the great thing about simulations is that you can do vastly more experiments. Because we have this single cell clock, we can actually use it in the simulation. So we do an experiment, we can say, did the simulation rejuvenate, or did it age the cell? So what I'm showing you here is we did 250,000 experiments in the simulation. So in each experiment, we're overexpressing 2 genes. So we're basically saying, look, if we increase the level of these transcription factors, these 2 transcription factors, does the cell rejuvenate? And this plot shows you the change in age in response to different combinations of genes. So you can see the vast majority of combinations age the cells, right? Most of the bar is to the right. But on the left, there's this really sort of exciting collection of genes, which is still about 1,000 genes, right? Or 1,000 combinations of 2 genes. But these are predicted to rejuvenate the cell. So what we're going to be doing in the new year is we're going to take the extremes. We take the combinations that are predicted to be most rejuvenative, the ones that are predicted to be most pro-aging. And there's 2 scenarios. One is we actually find things that are rejuvenative. Another more likely scenario is that actually our simulation's not quite there at the moment, but we've got the perfect data to retrain the simulation and make it better, and go again and go again. So it creates this positive feedback loop where we start to cover some real ground. So we're quite excited about what this might tell us in the new year. There's just a couple of ways to simulate cells. One is called GEARS from the Leskovec lab at Stanford. The other is called SCGPT, which is from Bo Wang at University of Toronto, so like using the transformer models that you might have heard of from ChatGPT. There's pros and cons to each one. So GEARS seems to be slightly more accurate. SCGPT seems to be better in response to gold standard interventions like rapamycin, Yamanaka factors. And we think there's a higher upside to working on these transformer models, so this is something we're going to be looking at. So that's use 1, right? So map this landscape and find the best possible start point for a drug that rejuvenates cells. The same biology seems to reverse disease, which is this really nice windfall. It helps us develop drugs for disease that's out there at the moment. So use 2, there's about sort of over 3,000 drugs that are clinically approved we can just get hold of, and we can run the clock across all of these drugs and just ask the question, do any of these clinically approved drugs rejuvenate? So this is something we look forward to doing again next year. And then lastly, this is a diagram showing you, again, individual dots are individual cells, and they're grouped together by their similarity in gene expression. So I'm showing you a time course, day 0, day 1 to 5, all the way around to day 21. Basically, these cells are receiving Yamanaka factors and converting into stem cells. And on your right-hand side, no, your left-hand side, this is the dangerous cancer-causing pathway, and then on your right-hand side, it's the age of the cells according to our clock, and you'll notice between day 1 to 5, or that clump of cells, and day 9, you see this difference in age, even though there isn't any change in sort of cell state to become a stem cell. So we sort of lock in on the rejuvenation event, and now we're going to study that more closely. We moved labs recently, so we were actually at the Gurdon Institute. So John Gurdon was a Nobel Prize winner along with Shinya Yamanaka for the discovery of cellular reprogramming. Some weird poetry going on that we're at an institute that's involved in this biology a long time ago, and we're now in the lab. We actually have no connection to John Gurdon, but there's space in that lab, so very excited. And I think I moved house and lab at the same time, so this is a recovery trip. Giving a presentation to you is a type of therapy for me. And then this is just a little bit of a tangent, but like, what are the personal motivations for me as a scientist to sort of pursue this doggedly? Because this is a very long road ahead. You know, this is very early stage science, and eventually we want to get to one part of the body and then across the body. So what keeps you going? And the first thing that keeps you going is we haven't got very long. So this is a chart put together by Tim Urban from the blog Wait But Why. And each square is a week of your life, so 52 squares across, 90 squares down if you're lucky. I'm at about 37, so I'm well on my way. But there aren't that many squares, right? Like when you look at this, there aren't that many squares. So, you know, if you love life, if you love specifically healthy life, and you want more of that, like we need to crack on with this problem. There's no reason for complacency. Like we need to be going as fast as possible, given that we're within reach. Wouldn't it be tragic if we were within reach and we just didn't sort of go fast enough? So that's sort of the stick, right? You haven't got that long. But I'm just going to talk about the carrot, and I'm sure people will tear me to pieces later, I don't mind. But I call this the great delta of time and space. So I'm not a cosmologist, but you might have seen this diagram, which is sort of the history of the universe. So Big Bang at the beginning, sort of an initial expansion, a lot of time, accelerating expansion of the universe. So at the moment, what is our experience? We're born out of nowhere on this rock, just one point in space. 100 years, we're gone. Like, what on earth's going on? How many layers of reality do we not have perception of? The whole thing's pretty crazy. When you have biological age control across the body, the forward-facing future looks very different. So, of course, you can have more time, but with more time, you can actually make the journey across space. So this sort of delta of time and space opens up in front of you, and the entirety of the future eventually becomes synonymous with your future. And that's just such an amazing forward-facing view, as opposed to just suppressing any thought of death, which is absolutely terrible. It's sort of unspeakable at funerals how bad it is. You have to sort of suppress it. Wouldn't it be nice to sort of lift that weight up and then have this forward-facing view, which is this sort of massive menu that we've never had before? So I sort of, this is very exciting to me. I don't want to seem ungrateful for a long and healthy life, which is what everybody expects at the moment. In the UK, we have a term which is a good innings. You had a good innings. And so, I'd be grateful for a good innings myself, but this is within reach, and I find this quite exciting, especially with what's going on with SpaceX and all this infrastructure sides sort of opening up. So I hope you guys don't mind the tangent. And then just the credits, I get to come here and talk about all the interesting stuff, but all of the really hard, difficult, careful work is done back home by the team. So we've got a team of 9. Andreas, Raya, Steve, Charlotte, Kate, Samira, Brendan and Lucas. Lucas talked earlier this week. Our investors, Jonathan, Milner, F-Prime and Kindred, and our advisors, Tony Calzarides and Leo Wozniak. And this image on the right was actually generated by the visual function on ChatGPT. So I think Lucas typed in cellular aging clock with DNA, and it spits this out, which I think is kind of amazing. I think it's got some of the numbers wrong, but apart from that, I couldn't make a better picture. So it's we've got somewhat of an affection for it, even though it was artificially generated. Thanks very much for listening. Any questions?
Hey, super cool presentation. When you showed your picture of your experiments, the combinations of 2, right? You had like a lot of combinations that age between 5 and 10 years or something like that. But your rejuvenation on that scale seemed only like a few months. Was the scale like the same in years, or why do you think that the rejuvenation seems to have been shown so much less?
Yeah, so there's some peculiar features of the data that we use to sort of train the models. So the data that's trained to train the models, the cells actually undergo like this mini electrocution before we measure the age. It's just how we get genes into cells. We basically have to electrocute them. And then the genes go in, they overexpress. We think that electrocution is basically aging all of the cells in this system. So that shift to the right, we think is mostly artificial. If we use a slightly different cell simulation, it looks more, when you eyeball it, it makes more sense. It's like a normal distribution. Most of the cells seem to be at 0 for whatever reason, and then you sort of have a few that are rejuvenative and a few that are very pro-aging. But the really exciting thing is, and you've got to be careful because it can be a mirage at the beginning when your simulations are basically as bad as they're ever going to be, but we do have combinations of genes that are predicted to be as rejuvenating as the Yamanaka factors with no pluripotency. So we'll see, right? We've got to keep going because it's very easy to get excited about that scenario, but the more likely scenario is that we've got to keep improving that simulation, but it won't take too long. It won't take too long, yeah.
Right here. Dr. Sinclair lab did the rejuvenation of the mice nerve, and it was criticized by Dr. Charles Brenner. And I don't know if you're familiar with the conversation. And we offered them in our university to duplicate the, his criticism of the mice. And he said he didn't have time for it. We was just chatting over the Twitter. Have you done mice or any other experiment that did produce such result as Dr. Sinclair's? And if you are interested, we can offer you the same experiment lab.
Yeah, very good question. So David Sinclair has actually used the Yamanaka factors minus the oncogene, CMYC, to reverse blindness in different models. So there's a glaucoma model, laser-induced blindness, and he deserves a lot of credit because he had quite a dramatic effect. So, funnily enough, that's the route that we want to go as well. And there's various factors that influence that decision. But we can go with a gene therapy, for instance, and blindness is very binary, so it's a very good endpoint. But it is something we're looking at. We're at a very early stage. We're just trying to find the right combination of genes, but essentially we're going to go down the same path. Yeah.
Hi, thank you for your talk. In one of your slides, you were talking about the different models that people have constructed to look at modeling cells. I forget the names, but you showed the significance. And you also talked about the model that you're constructing and how you've collected all these different samples. And you mentioned to train your model, you were using something on the order of 60 samples, and then evaluating your training data set with something like 40. I'm a little concerned that that's small, and how do you know that that's actually representative of the nature of fibroblasts? You can think about them being isolated from different organs. They have different properties, and then these AI systems, building a neural network, for example, you usually want thousands of examples. So how do you justify the small data set being representative of the biology itself? Thank you.
Yeah, so with the data set, we actually it was like a rolling construction. It was like, look, how much do we need? And like how much extra are we getting from each additional samples? But it turns out sort of 50 to 60 samples is way more than we need. We've sort of reached the top of the S-curve. The reason being we're doing single cell analysis. So from each of those samples, we're actually taking like 500,000 cells. So together, it's a lot of cells. The sample just gives us the chronological time label, so that's important. But actually, it's a vast number of cells. We're almost annoyed that we spent the extra money because we could use that for the next bit, but it's better to have too much. I'm quite happy to sort of talk about the specifics in follow-up if you want to sort of go into the various aspects. And then you talked about the model that we use.
You were constructing the model, and I would agree with the single cell provides a lot of data. Yep. But then those are 61 samples from, I'm assuming, 61 unique individuals. That might be a lot of data per individual, but that's not really representative of the population, and you can correct me if I'm wrong.
Yeah, so just yeah, sort of in the interest of time. So
Sure.
yeah, we tried to keep it as representative as possible because originally we were like just working with 52-year-old female fibroblasts. We were worried that we were only ever going to create a therapy for 52-year-old females. So we did a good age range. It was the gender split was pretty even. We had African American samples just in case there was some variation, and the clock behaves very well across all of that sort of minimal scope of variability. But the moment we find something in fibroblasts, some really important questions. Even if it's safe in fibroblasts, is it safe in another cell type? Does it even work in another cell type? So I'm going to try multiple different cell types. So it's like layers of the onion. Like we just got to go forward step by step. As, our simulations aren't perfect as well, so we've got to be very careful about false positives at an early stage. Let me talk to you afterwards. Yeah.
I appreciate it. Thank you for the talk.
You're welcome.